Hi everyone, this is the second lecture in chapter 22. In this one I will be discussing microorganisms in water for human use. So we're going to start out with the purification of water for drinking. So drinking water mostly comes from rivers, aquifers, and springs, so natural water sources along with aquifers. Sometimes we have reservoirs too. And then only in remote or underdeveloped areas or high mountain areas is water used straight from its natural source. In most cities, it has to be treated when it is supplied to consumers, so supplied to our houses or businesses. And the water supplies such deep wells that they're really clean and free of contaminants, so they require less treatment for those that are laden with waste, so something that would actually be recycled from a home. So when we talk about that wastewater, then we need to get into sewage treatment. And sewage is an interesting product because it contains large amounts of different things, including solid waste, dissolved organic matter, and possibly toxic chemicals that could pose a health risk. So in order to remove all of these health hazards, it typically is broken down into three phases. Our primary stage is going to separate out large matter, things that are solid waste or larger pieces of particulate. A secondary stage would then reduce the remaining matter and remove some of the toxic substances. And then that third stage is where we have the completion of water purification. So let's go into these three phases of the water treatment a little bit more. When we talk about that primary phase, remember this is where we have the solids taken out. So in that primary phase, the floating of bulkier materials, those will be skimmed off, and some of the smaller suspended particles that don't rise to the top will be allowed to settle to the bottom. In our secondary phase, we use a huge diverse community of natural bioremediators that can aerobically decompose the remaining particles. Okay, so bioremediators should tell you that they're biological mediators, right? And then that tertiary phase is going to include filtering and often chlorination prior to discharge. So generally speaking, when we talk about reclaimed sewage water, it's usually used for watering golf courses and parks rather than drinking, although it may be gradually released into larger bodies of water where the dilution of that would then allow it to be able to go through the process to be drinking water. And scientists are working on and have found ways to harness energy from bacteria found in sewage. Many of the wastewater bacteria will form biofilms on rods that can be inserted into the sewage, and those biofilms can generate electrons, and then we can transfer that using copper wires to electrodes producing electricity. Now it doesn't produce a lot yet, but the hope is that perhaps in the future that is a way of getting energy. So let's look at the water treatment in a big picture kind of view and look at the major steps in both water purification and sewage treatment. So this is figure 22.6 in your book, and it has the major steps in water purification. So starting over here at the left with number 1, we have impoundment or taking in of the water into a large reservoir that serves two purposes. It's storage. We're going to store water there so it's not just that river running through. In addition, we're going to have sedimentation. Things will drop to the bottom, settle to the bottom, okay? And then next, that water is going to be pumped into a holding tank where aeration takes place. That means air being put into that, okay? And it will be stirred here. From this, we will go to the third basin here that has coal and sand on the bottom two layers of it. And we expect to have further settling and filtration taking place in this holding tank. From this, we'll go along this pipe with chlorine being added, and the chemical disinfection is done with that chlorine gas bubbling through the tank, and the treated water is kept in a storage tank here. This then will travel up, and it will go through the consumer, into the consumer's house through domestic water pipes, okay? Now this starts a whole nother round of, we then have wastewater out of our home going to a wastewater treatment plant. You'll see here we have what happens at that wastewater treatment plant. Starts out with raw sewage, keeping that liquid residue above what settles or is skimmed off. We have the solid waste going one way, that liquid then coming down. Here we're going to have the sludge digester with the water being aerated and mixed. It's going to go through filtration. Some settled solids here will be removed, and it's aerated here, and it will go down and be filtered. We will have solids coming out. We will also have treated sewage released into a body of water after chlorination. And this is really treated sewer. It's really, most of the things have been removed from that. Okay, and so we'll go back to the body of water, and eventually that water will make it back here, and we go through the whole step again. I have included a video in this module that you can see on a sewage treatment plant. So that's kind of what we do with water purification, but how do we monitor water and prevent diseases? We all kind of take that for granted, that we can just get water out of our tap and have clean water. So when we talk about the microbiology of drinking water, in the U.S. we're very lucky, we have very clean and sanitary water, but worldwide epidemics of cholera have killed thousands of people, and it is a lack of sanitary water that is responsible for billions of cases of diarrheal diseases. So think about that, billions of cases per year that can kill up to 3 million children a year. And that is just the microbiology of drinking water. In the U.S. even, we have nearly 1 million people that develop waterborne illness every year. This generally isn't obviously from tap water, but getting it from other sources. And good health is dependent on clean, potable water. It has to be free not only of pathogens, but of toxins, also any disagreeable turbidity, things floating in it, odor, color, and taste. So the things that we're most looking out for, or the most prominent waterborne pathogens, fall into some different categories. Out of the protozoa, we have Giardia and Cryptosporidium. Giardia is one that you often hear about people getting from stream water if they're camping or something. We also can have bacterial pathogens. Campylobacter, Salmonella, Shigella, Vibrio, and Mycobacterium are the most prominent ones. And then we can also have waterborne pathogens that are viruses, and the most prominent ones are hepatitis A and Norwalk viruses. So some of these agents can actually survive in natural waters. Giardia is a very good example, for long periods without a human host. Some of the other ones are present only transiently and are rapidly lost. So when we're looking at the microbial content of drinking water, it has to be continuously monitored to make sure that it's free of infectious diseases. So how do we go about doing this? It's difficult and really time-consuming if we were just to be testing for specific pathogens. For example, if we were just looking for Giardia, it would be very difficult to do it for one particular pathogen. So what is generally done is water purity tests are focused on detecting any kind of fecal contamination, because that's where many of these pathogens would be found, is in feces that might have come from livestock, something in that order. So if there's a high fecal level, it can mean that the water contains pathogens and is not safe to drink. So when we look at this, we sometimes look for indicator bacteria that would show that there is fecal contamination. So there are intestinal residents of birds and mammals that can be easily identified using routine lab procedures. And the ones that we look for are coliforms and enteric streptococci. And the reason why we do that is that they're great indicators of a high level of fecal contamination. They're very commonly found in fecal contamination. So the coliforms that we look for are generally gram-negative, lactose fermenting, and gas-producing bacteria. The main ones would be E. coli, Enterobacter, and Citrobacter. And it's actually the EPA standards for water sanitation that are based on these primary levels of coliforms. We're looking for these coliforms. So we have actually been using the same coliform assay as a standard procedure since 1914. And it's a coliform count, and it's used to regulate both food production and to trace the causes of food-borne outbreaks. But recently, there have been serious problems that we've noted with this use of coliforms to indicate fecal contamination. And the main things are that other species besides E. coli are commonly found in non-fecal environments such as fresh water and plants that can end up in our food. So we could have a false positive because the E. coli may have come from something other than a fecal environment. So what we're finding is that maybe this is not our best method, because unless we're looking specifically for E. coli, you can't be sure then that it's feces. So the problem is that we're doing kind of too non-specific of a test. So currently they're looking at using different assays, but this is still the one that we're using. And it may just be that we're getting lots of extra false positives. So that's the end of our water and sewage treatment and then drinking water lecture. There will be at least one more lecture on this chapter. Okay, thanks.